Assalamu alaikum everyone. Continuing from where we last left, this might be a relatively long episode. So starting with this simple microscopes, the first type of the four types of microscopes. These microscopes have a single magnifying lens, as the one used by Leeuwenhoek. Not much to say about this, so let's continue. Compound light microscopes. Compound because they have multiple magnifying lenses, light because they use visible light as a source of illumination. They were first developed by Hans Janssen and his son Zacharias. Modern compound light microscopes have the typical shape that you all know. We use compound light microscopes, especially the bright field microscope, in schools and universities for learning purposes. Modern compound light microscopes types are bright field microscope, dark field microscope, phase contrast microscope, and fluorescence microscope. So starting with the bright field microscope, this is the easiest one to use. It has a bright background, and we can use it for stained specimens or unstained specimens. It's better to use it for stained specimens because the background is white and the unstained specimens will appear white also. So there will be not much contrast for using it for unstained specimens, but we might use it for unstained because staining the specimens will kill it. So we don't have stained and living specimen. So it's either stained killed or unstained living. This one and this one are micrographs for a bright field microscopy. So these grape-like clusters are clusters of blue-stained Staphylococcus aureus bacteria in a blood film. While this one, you can see clearly that it has the specimen is white, so it's unstained, and we have poor contrast. The dark field microscopy uses a dark background, so we don't need the staining. So the specimen will appear white or illuminated against a dark background. So we use it for unstained and living specimens. We especially use it for thin spirosheets. Thin spirosheets, we cannot view them using the bright field microscopy, but we can view them on a dark field microscopy. Thin spirosheets like Treponema pallidum, the cause of syphilis. This is a Treponema pallidum bacteria. It has a spiral shape, and you can clearly see it against a dark background. Okay, the third type is a phase contrast microscopy. This one is tricky and has a complex mechanism, but you are not required to exactly understand how it acts. But in general, it uses the phenomenon of wave interference of light, where a special condenser annulus and a phase plate are used to cause constructive and destructive interferences of light waves passing through structures of different density. So structures in specimens that are different in density will have different refraction of light, and the phase plate magnify that effect to cause constructive and destructive interferences. The constructive will cause these structures to become more lighted and more shined, while destructive interferences will make them appear darker. This helps us distinguishing cellular structures from each other by contrast, according to their differences in density. So this allows the viewing of internal structures in a living specimen without the need for stains. I will put in the video description below a link to a video that might help you understand the mechanism of phase contrast microscopes. So this is a micrograph of a phase contrast the same cells appear more distinct and more contrasted. So this is a good contrast. Okay, so the fourth type is a fluorescence microscope. It uses the phenomenon of fluorescence. So what is fluorescence? Fluorescence, we have materials um, that when excited with UV light, they emit a visible light. So UV light is invisible. So when we target them with UV light, they reflect visible light, a light of longer wavelength. So these are called stains or dyes, and we use them to stain specimens. So they use ultraviolet light instead of visible light as the source of illumination, and they view stained specimens against dark background. So these are the same Treponema pallidum species, but they appear green on a dark background. How could we stain only the Treponema pallidum bacteria, but not the rest of the specimen? We use anti-Treponema pallidum antibodies, and we attach the stain or the dye, the fluorescent dye, to that antibody. Okay, so when we put the dye on the specimen, the anti-Treponema pallidum antibodies only target the Treponema pallidum species, so the stain only stains the bacteria, but not the rest of this, the specimen. This is called immunofluorescence. So we can tag fluorescence stains, for example, fluorescein isothiocyanate, with immunoglobulins antibodies that bind specific cellular structures. So when we stain a specimen, only those specific structures or specific microbes shine. This is called immunofluorescence, and it is very important in labs as a diagnostic procedure. Fluorescence microscope is used to detect microbes in cells and tissues. There are two types of immunofluorescence. The direct immunofluorescence we have just used here. The stain is attached to antibodies that can attach to the thing that we are examining. For example, here the Treponema pallidum bacteria. But the indirect, so in autoimmune diseases, we have antibodies already attacking cellular structures. So what we do, we use antibodies that can attach to those antibodies that are already attached to the tissues in the specimen. So indirect immunofluorescence, antibody binds to another antibody that is already attached to a cellular structure. It is used in the diagnosis of autoimmune diseases. Okay. Now we will talk about the parts, the structures, the components of modern compound light microscope. So starting from below, we have the base and the arm, and these structures help us in holding all the structures of compound light microscope. When we bring the microscope by our hands, we have one hand grabbing the arm and the other hand grabbing the base. So in the base we have the light source, we have the on-off switch for that light source, then we have the rheostat control knob that controls that increase or decrease the intensity of light produced by that light source. So the light source goes up through all these structures, reaching the eyepiece, then your eyes. Okay, so firstly, the light passes through the field diaphragm that has a collector lens. The collector lens helps make the light waves parallel, so they are produced divergent. They go here like this and like this. Then the collector help them go parallel to each other to reach the condenser. We have the field diaphragm lever that can control the light passing through the field diaphragm and the collector lens. So there is another way to control the light other than the rheostat control knob, which is the field diaphragm lever. Okay, so the light reached the condenser. Condenser help the light focus on the specimen. Okay, so it helps the light focus on the specimen. So we have the condenser and we have the iris diaphragm control arm that control the light passing through the condenser. So this is the third way of controlling the light intensity. Okay, so we have the condenser control knob that can control the height of the condenser. We can make it go up or down. Then the light reach the stage which has this specimen. Okay, so this is the coarse adjustment knob and the fine adjustment knob. These structures help us elevate or decrease the elevation, the height of the stage. The coarse move the stage rapidly. The fine knob moves it slightly. This helps focusing by the objective lens. So the light hit the specimen, then went to the objective lenses. Objective lenses are the magnifying lens that is magnifying the specimen. Okay, so the first lens that is magnifying the specimen is the objective lens. Objective lenses revolve or turn around the revolving nose piece, so they can switch places. We have 4 types of objective lenses, from scanning objective to oil immersion objective. I will talk about them in a second. So revolving nose piece holds the objective lenses. Then we have the binocular body that holds the eyepiece. The eyepiece has a magnifying lens that magnify 10 times. So the magnification power of an eyepiece is 10 times. Did we miss anything? I don't think so. Okay. This is a summary of what we have just said. You can go on them by your own. Okay, so the magnification of the compound light microscope can reach up to 1000. How is that possible? As I just said, we have 2 magnifying lenses, the objective lens and the ocular lens. The objective lens are two types. We have the scanning objective that can magnify by 4 times, the low power objective that can magnify by 10 times, the high dry objective that can magnify 40 times, and lastly the oil immersion objective that can magnify by 100 times. So each one of these objectives, in order to calculate the total magnification by a compound light microscope, we multiply that by a 10. That is for the ocular lens. So 4 by 10 is 40, 10 by 10 is 100, 40 by 10 is 400, and 100 by 10 is 1000. The higher the magnification, the more light we need. Why do we use oil? Because oil has the same refractive index of glass, so light passes through the specimen, then the oil immersion objective with minimal refraction. This helps improve the magnification power and resolution, achieving a 1000 magnification power. Okay, moving on to electron microscopes. Electron microscopes use an electron beam instead of a light source as a source of illumination. So this is a diagram of a scanning electron microscopy, and this is a diagram for transmission electron microscopy. So both of them have electron gun at the top that is placed in a column, in a long column like this. Then we have at the bottom a chamber that is void of air. It contains only a vacuum, and it contains the specimen like here and here, and then the specimen like here and here, and then an imaging technique, a camera system inside it. Okay, so here and here. So the specimens in electron microscopy, scanning and transmission, are killed, always killed. Why is that? Because living creatures cannot sustain the vacuum. Also, the specimen processing techniques, the complex processing techniques in electron microscopes always kills the specimen. Okay, so the transmission electron microscope, okay, what it does, it produces electron beam that passes through magnetic magnets that can focus it onto the specimen. Then these electrons hit electrons in the specimen and generate secondary electrons that are knocked out from the specimen. So the secondary electrons are off the specimen and they reflect on the secondary electron detector, which is the camera system inside the scanning electron microscope. The scanning electron microscope is called scanning because it scans the specimen point by point, and it scans the surface of the specimen point by point, and each time the secondary electron detector detects how much electrons are knocked out and reflected from the specimen point by point, and it uses that information to produce an image of that specimen, of that surface of the specimen. We have the scanning electron microscope. This is a mosquito. You can see clearly how much detail of the surface of the mosquito we can see by the scanning electron microscope. And this is the grape clusters of Staphylococcus aureus. They really look like grapes. And this is how it looks like in real life, the scanning electron microscope. Okay, did we miss anything? Okay, so they can't view living specimens because of the vacuum specimen processing. The images are okay. The images are always, for all electron microscopes, are in black and white, but we can artificially color them later. Okay, we said all that. Scanning electron microscope is used to view the shapes and outer surface details of microorganisms, especially bacteria and viruses. Okay, so moving on to transmission electron microscope. It uses a similar strategy. So we have the electron gun at the top, producing electron beam that can pass through the specimen. The electrons pass through the specimen. Some of them pass and some of them get blocked. Those that are passed then get focused on the imaging plate and produce an image of that specimen. So they, instead of reflecting off the specimen, they pass through that. And this is the transmission electron microscope mechanism. So transmission electron microscope has higher resolution and higher magnification power, as we said in the previous episode, and it helps us study the internal structures of viruses and any other microbes. So this is a transmission electron micrograph of Staph aureus bacteria in a binary fission. You can see different stages of binary fission, and here is the transmission electron micrograph of influenza A virus, and here is what it looks like in real life, the transmission electron microscope. Okay, did we miss anything? Okay, it uses electron beam that pass through a very thin specimen. Some electron pass while others are blocked, so an image is created and displayed on a monitor. TEM enables scientists to study internal cellular structures. Okay, lastly, we have the atomic force microscope. This is the newest technology. There is a silicon cantilever with a sharp tip. It's called probe, and we use that to physically scan the surface of the sample. The sample is living and in full physiological conditions, and we just pass that cantilever and tip on its surface without causing much distress to the specimen. Okay, and we form a 3D image of that. How we do that? While the cantilever and tip passing through the surface of the sample, atomic forces between the specimen and the tip causes the cantilever to deflect and oscillate slightly. So we use a laser beam to magnify that effect on a photodiode, which translate that into a detector that can create an image of the surface of the specimen. So AFM allows us to observe living cells and microorganisms at extremely high magnification and resolution under physiological conditions without stains to produce a 3D images. So some of you might say atomic force microscope is very similar to scanning electron microscope. Both of them scan the surface of microorganisms and small things. But really the difference is atomic force microscope view such small structures under their normal physiological conditions, and they are living without stains. So that's the really helpful thing in atomic force microscope. This is the last slide of this chapter. It is a summary of what we have just said about compound light microscopes and electron microscopes. So compound light microscopes are 4 types: bright field, dark field, phase contrast, and fluorescence. All of them have a resolving power of 0.2 micrometer and a useful magnification of 1000 times. And bright field is used to observe morphology of microorganisms such as bacteria, protozoa, fungi, and algae in living, the unstained, and non-living, the stained state. Objects are observed against a bright field background and cannot observe microbes lesser than 0.2 micrometer in diameter or thickness, such as spirosheets and viruses. The dark field can observe thin spirosheets, so useful for examining thin spirosheets like Treponema pallidum, and they view unstained living organisms against a dark background. They are slightly more difficult to operate than bright field. The phase contrast can be used to observe unstained living microorganisms, just like the dark field. And lastly, the fluorescent dye attached to organism, primarily an immunodiagnostic technique, immunofluorescence, used to detect microbes in cells, tissues, and clinical specimens. Transmission electron microscope has a resolving power of 0.2 nanometer and a useful magnification of 200,000 times. Characteristics: the specimen is viewed on a screen, just like the scanning electron microscope. The transmission electron microscope has excellent resolution, has the highest resolution of all the microscopes. It allows examination of cellular and viral ultrastructure, the internal structures, and the specimen is non-living, just like the scanning electron microscope, and reveals internal features of thin specimens. The scanning electron microscope has a resolving power of 20 nanometers and a useful magnification of 10,000 times. The specimen is viewed on a screen. The specimen is non-living. It gives the illusion of depth, 3 dimensions. It is useful for examining surface features of cells and viruses, and resolution is less than that of transmission electron microscope. Thank you for listening. Please like, comment, and subscribe, and be tuned for the 3rd chapter in the next episode. Thank you for watching.